Suyash Shringarpure is a senior scientist and statistical geneticist at 23andMe, and he is developing machine learning and statistical methods for analyzing 23andMe's large genomic and phenotypic data set. So I thank you very much for making the time to come to us. He is coming to us from Sunnyvale, California. And I want to welcome you, and I'll turn this over to you now.
Thanks, Chris. Let me share my slides. All right. Let me know if you guys can see my screen.
I can see it. Yes, it looks good.
Perfect. So good morning to all of you from Sunnyvale, California. My name is Suyash Shringarpure. I'm a senior scientist in statistical genetics at 23andMe. And today I'm going to tell you a little bit more about rare disease genetics research and some of the work we've been doing using self-reported medical data that we have. Okay, so I think I keep this slide in my presentations for just for context for many people, but to this audience, it's probably not necessary. We know that there are about 30 million people in the US affected by rare diseases overall, even though each rare disease affects a small number of people. And only 5% of rare diseases have approved treatments, even though we think about 60 to 70% of them have known genetic causes, and we know about the genetics of maybe 40 to 50% of them. So on the sort of rare disease side, that's a great reason to study them, to try and figure out whether there are new therapies we can develop. But there are also connections between rare diseases and common diseases. And there are interesting scientific questions there about what can we learn from rare disease genetics about the genetics of common diseases that can also help us understand those other diseases. So today, I'm going to give you a very brief overview of rare disease genetics research. That's probably a multi-hour topic of its own, but we'll cover it in a few minutes. And then I'll talk a little bit more about how we've been using self-reported medical data for studying rare disease genetics. And almost everything that I'll talk about in the second part, we have a preprint on medRxiv. The URL is at the bottom, and you can look for more details if you're interested about that study there. Okay, so rare disease genetics currently is studied primarily through whole genome sequencing or whole exome sequencing, which is sequencing of just the protein coding parts of the genome. And this figure from a 2011 review paper shows a few ways of commonly studying rare diseases. So in panel A, where you can see these three different family structures, the parents are on the top connected by a line, and then a child shown by this circle here. Three different disease cases from unrelated families are whole genome sequenced. And then if they have the same rare disease, then the hypothesis that maybe all these three individuals carry some genetic variation in a specific gene that is likely associated with having this disease. And so by looking at their genomes and looking for common genes which have variation, you can try to identify which genes are causing the disease. And this is helped by now having large panels of whole genome sequence data from populations across the world. So you can hypothesize that variation that is common across the world in multiple populations is not likely to cause a rare disease. Because if it were causal, then many more people should have the disease. Sort of one identifying genes associated with rare disease. The panel B, which shows now, instead of just a single mother, father, child trio, it's showing you a whole large pedigree of a family where the blue squares indicate affected individuals. This is the slightly older paradigm of studying rare disease, where before whole genome sequencing was very popular, they would look for these large pedigrees where multiple family members are affected by disease and ask which regions of their genomes are shared between these affected individuals. Of course, because these are all family members, they will inherit some parts of the genomes from their ancestors, and they will all share some parts of the genome, but you can look for parts that are shared more frequently in affected family members than unaffected family members. A third design is where you sequence a single mother, father, and an affected child. And here, both the mother and father are assumed to be not affected by the rare disease. And then you can hypothesize that since the mother and father don't have uh, the rare disease, maybe the child has the rare disease because they have some novel genetic variation that may be causing this rare disease. And lastly, when the underlying the disease characteristic is an unusually high level of, say, an enzyme or a protein, then you can look at the population distribution, say for cholesterol, and ask, are there people who have really extreme levels of cholesterol? And maybe they are likely to have, say, a rare form of high cholesterol called familial hypercholesterolemia. So these approaches have been successful in increasing the number of rare disease genetic discoveries. On the right is a figure showing how many genes have been discovered by these methods. And so here the red indicates the conventional family-based approach of gene discovery, and blue indicates the next generation sequencing-based approach of rare discovery. And you can see compared to 1986, the number of rare diseases discovered per year has been increasing over time. The limitation of this approach is that this primarily works only for Mendelian diseases, where a defect in a single gene can cause the disease. And there could be multiple genes that cause the same disease, but having a genetic variation in any one of them is enough to cause the disease. And this is, of course, constrained by sample size. You need to be able to find the 5 or so unrelated cases that you need to confidently identify what the shared genetic variation might be. A slightly different way of doing rare disease gene discovery for a different class of diseases. So just as some diseases can be Mendelian or caused by a single gene, there can be rare diseases that are complex and caused by multiple genes, each gene contributing a small amount of risk, but together they're enough to cause the rare disease. So these polygenic rare diseases are studied through genome-wide association studies, and I'll spend a minute on that figure on the right just to give you a sense of that. So at the highest level, it involves comparing the DNA of many affected individuals to unaffected individuals. So affected individuals are called cases, unaffected are called controls. And by assuming that everything else about them is the same, except for their disease diagnosis, you can claim that any gene region that is more common in the cases compared to the controls is likely to be associated with the disease. So this is, on the right, is a standard way of representing results from such a genome-wide association study. So here in the figure on the x-axis are shown all the individual chromosomes from 1 to 22. And on the y-axis is a measure of statistical significance, the p-value, but transformed slightly so that it is a higher number means more statistical significance and a stronger association. And in the figure, the words on the top highlight all the genes where the genome-wide association study is finding statistically significant associations. And by looking at the function of some of these, you can try and understand what genetic pathways or what bodily organ systems are involved in the disease. This has worked quite well in common diseases where multiple small contributions from genes lead to disease, and also in some rare diseases where we have enough sample sizes. And that's one of the limitations of this approach, which is because this is looking for small effects, it does require that there be large sample sizes for discovery. So I'll give you a brief introduction to 23andMe research in case you're not familiar. So you may be familiar with 23andMe as an ancestry genealogical related research tool. So what you can do is you can order a 23andMe kit, you can spit everyone, and you send it through the mail, and you get your results about what your genetic ancestry is like, who your relatives might be. But we also have a research component where everyone who becomes a customer is asked, do you want to opt in to have your data be used for research? And we have a IRB approved protocol and consent form. And our protocol is not specific disease based, it's broadly for many diseases. And because it's participation at any time and resume at any time. And the data we have is all aggregated and anonymized. So no one has access to sensitive customer details like, say, names and addresses. All we have is anonymized genetic and phenotypic data. And this model is quite effective for research because participation is easy. People can just sign up online. They can contribute data through online surveys. And as a result, geography is not a barrier. We can have people from all over the world participate in this research. And another advantage is that every person can be in multiple studies. They don't have to be in one study at a time. So we've used this model for studying many common traits and diseases. So as an example, here is a genome-wide association study using self-reported data for morningness. So we asked people, are you a morning person? And they could answer yes or no. So we said people who answered yes are cases and people who answered no are controls. Here on the top is a genome-wide association study from a few years ago where we had 39,000 people who said yes, 50,000 people who said no. And in this Manhattan plot, things colored in red are the signals that are statistically significant above a threshold. And you can see there's a small number of signals here in the top. Many of these are actually genes that are known to be associated with your chronotype or circadian rhythm, but there are a few signals in this. But at the bottom, as time passed and many more people took the survey and answered the question, we now had 250,000 cases and 220,000 controls. And you can see in this figure that we have many more genetic associations for morningness now. And so this, like I mentioned, for GWAS, increasing sample size allows you to make more discoveries. So the 23andMe cohort that we have now is well suited for study of rare diseases, since we now have 11 million plus genotyped customers, which means that something that has a disease frequency of 1 in 10,000, we will still have hundreds of cases for. 80% of genotyped customers consent to research, and about 30,000 surveys are completed per day. So we asked, can we use this model that we have for studying common diseases and traits and study rare diseases instead? So that's what I'm going to talk about next. And like I mentioned, we set this up as a data collection using self-reported diagnosis and then analysis by GWAS. So here's an example of how it works for studying cystic fibrosis. So we developed a rare disease survey which had 7,000 rare diseases in it, and someone could type in the name of their rare disease, and we had an autocomplete option that would allow them to choose their rare disease. And at the bottom, we have a second survey that everyone answers in the beginning called the health history survey. And so the health history survey has yes/no questions that ask something like, have you been diagnosed with cystic fibrosis? So in the rare disease survey, if someone had cystic fibrosis, they would start typing in cystic fibrosis, it would autocomplete, and we would tag them as a potential case. Similarly, if they answered in the health history survey that they have been diagnosed with cystic fibrosis, we would tag them as a case. But if in both surveys they said, I don't have a rare disease, and in the health history survey they answered no to the cystic fibrosis question, then we'll call them unaffected and tag them as controls. And then by comparing the genotype data of both these groups together, we can get a signal like a genome-wide association study like on the right here. And here you see there is only one statistically significant signal that we find. It's around the CFTR gene that is well known to be associated with cystic fibrosis. So this is not a novel result, but a good confirmation that this approach works. So we validated this approach through trying to rediscover known associations. We tried 33 rare diseases where we knew of rare disease association through one of these conventional approaches of sequencing and such. And for those 33 rare diseases, we found 29 known associations. So that made us confident that this approach works. And some examples of where these were found were cystic fibrosis, Lynch syndrome, beta thalassemia, cleft lip, and so on. And one example I can highlight here is in Huntington's disease, where the Huntington gene, so this is a zoomed in version of the same Manhattan plot I showed you before from the GWAS. And this is now focused on a small region on chromosome 4, which contains the Huntington gene that's responsible for Huntington's disease. And so here we have a signal that is just above the line that we consider the threshold for significance. And the genetic variation that we find that is significantly associated is one that is known to be common in Huntington's cases through sequencing as well. And as a more quantitative validation, when we looked at clinical GWASs that people had published before, we found that the estimates of effect size that we obtained from our GWAS on self-reported data were comparable to those that people obtained from clinical cohorts, suggesting that there might not be too much bias in our data compared to clinical data. So I'll show you a couple of examples, maybe not all the ones that I have, but here is vestibular schwannoma, which is a rare disease that is related to cancers, but is a benign tumor that usually develops on your on the nerves in your auditory system, on the vestibular or the cochlear nerve. And here we find one genome-wide significant association. It's near two genes called the CDKN2A and 2B genes. And if you look at what is known in the literature about these genes, on the top are associations of this gene with 5 different germline cancers, or cancers where this gene is associated in the germline variation setting. And on the bottom are another 5 cancers where somatic mutations in the CDKN2A, 2B genes are associated with known cancers. Yeah, the slides are stuck for a bit. So we have a couple of other similar examples we looked at, and in this case, to go back to the vestibular schwannoma, we can see that there are we have about 1,000 cases in our database, and this makes it the largest genetic study of vestibular schwannoma. The other example that I'll talk about in a minute once my slides start responding again is in an eye disease called Duane retraction syndrome that's associated with limited movement of eyes. And sorry, I'm going to stop my slides a bit.
We will have our producers pull up the slide deck on their end.
Actually, that would help. I'm
Okay.
not sure why the presentation is stuck. Thanks. Actually, can you move to the next slide, please? Yep. So this is the eye. So talking about it's an eye disorder where you have limited right-left movement of your eyes, and previously this was thought to be muscle related, but later found to be related to the nervous system. So you find one association that I've highlighted here in the middle. It's near 2 genes called OLIG1 and OLIG2. But the interesting part of this is the figure on the right. So this is a figure looking at nerve systems in mice, where people can actually do some of these experiments. And on the left part in panel A is showing how genes affect each other in a network or a pathway. So the gene on top called MAFP is known to cause Duane syndrome both in mice and humans. But below it is OLIG2, which is known to affect nerve development in mice, but has no known connections to nerve-related diseases in humans. So our GWAS is the first sign that OLIG2 may actually be associated with Duane retraction syndrome as well. So if you go to the next slide, we, as we like to do in statistics, we want to make sure that our results are robust. We try to ask, if you take an independent data set, can you find these same associations? We looked at UK Biobank, which is another large collection of about 500,000 British European individuals consented for research, and found that 3 out of the 6 associations that we had found novel replicate in the UK Biobank, and 5 out of 6 have an effect in the same direction, like increasing or decreasing risk. And if you go to the next slide, since sample size is still a constraint in these rare diseases, we asked, rather than just doing genome-wide association studies in one population at a time, what if we include data from multiple populations together? So use all individuals from all populations. And one limitation of GWAS I didn't mention is that closely related individuals can't be used in the analysis. But in this trans-ethnic analysis, we were able to include all individuals, including close relatives as well. And reassuringly, what we found is that these improve our statistical significance for all the associations we find, suggesting that the rare disease genetics is shared across populations, and studying it all together will enable us to make genetic discoveries faster. So to conclude, if you go to the next slide, we have a few next steps planned. One is, as more people complete these surveys, we can do more GWASs. And based on our results so far, we've found that for a few diseases where sample size is really the key bottleneck to finding new associations, we've started a new study, or started 2 new studies, one for idiopathic pulmonary fibrosis, one for systemic sclerosis, where we're trying to enroll additional rare disease cases. And this is based on, again, self-reported data. If you have, say, systemic sclerosis, you can go to this landing page that is described at the bottom. You have to fill in a few questions about your disease history and diagnosis, and then you can be enrolled in the study. So to conclude, on the next slide, we show that self-reported rare disease data can be combined with GWAS techniques to identify novel genetic associations. And an additional take-home here is that trans-ethnic analyses are really important for rare diseases, since sample size is the primary constraint, and it will increase our power to find associations. And lastly, that other biobanks, such as UK Biobank and all of us, will also enable more rare disease gene discovery. I'd like to end by just acknowledging my collaborators on the last slide. If you could. So my co-authors, and I would also like to thank all the research participants and employees of 23andMe for making this work possible. So I'd like to thank all of you for listening, and I'm happy to take some questions.
Okay, thank you very much for that great overview. We have time for a few questions. First is from our fellow Aaron Prater of the Gazette in Colorado Springs, Colorado. How common are polygenic rare diseases, or are they typically one-off cases?
No, there are actually, I guess I wouldn't be able to put an exact number on them, but of rare diseases, many more are Mendelian or monogenic, but there are at least polygenic rare diseases. I probably should not guess.
Okay. Another question from our fellow Miri Kim, a freelancer in the US. Can these GWAS results be used to find new or existing therapies for rare diseases that target these genes or pathways? Do you have plans for this in the future?
Yes, exactly. So that's one of the things we've been doing at 23andMe, asking, are these genetic discoveries able to therapy? And so that's our hope with these kind of diseases as well, that as we find more and more genetic associations, maybe some of these will be targetable by new therapies. And if we can develop them, that would be amazing.
Okay. Now, you studied 3 conditions, Duane retraction syndrome. I remember you tell us about that one. Vestibular schwannoma and spontaneous pneumothorax. Why did you start with those 3?
Yeah, I think that the theme is my talk has been sample size is really constrained in studying rare diseases. So these are actually conditions that are rare but not too rare. So in all of these, at least for vestibular schwannoma and spontaneous pneumothorax, we were actually able to assemble a data set containing more than 1,000 rare disease cases. This means we had good statistical power to find associations. Duane retraction, even though we had a really small number of cases, we had a signal that was statistically very robust, so we were confident in. And so that has been one of the filters that has gone into this, like asking, where do we think we have enough sample size in our data set to be able to confidently say this is a new discovery?
Okay, I think we have time for one more question. And this is from Marielba Nunez from SciDevNet in Venezuela. Have the patients any rights over the genetic information they share? And so I guess could you also just talk about what are the privacy protections that you put in place?
Yeah, so everyone who signs up for 23andMe has access to some genetic health reports. Our genetic health reports are potentially approved by the FDA, depending on the FDA's discretion. So those are a separate set. Data from research isn't directly shared with individual participants unless it becomes a report that is made available to customers. And privacy-wise, like I mentioned, on the research side, we don't have access to customer data. All we have is an anonymized numerical ID for everyone, and then we have their genetic and phenotypic data, so we don't know who's who.
Okay, and I want to thank you very much. I think we need to move on to the next session. It's been a great overview. And fellows, I just want to remind everybody that the slideshows will be available for you. So all the great detail that he has in there, you can go back and review it later to get some to make sure you're capturing all the new scientific nuances there. But Suyash, I want to thank you very much, and then we can bring on our next guest.